Sleep and wakefulness are two alternative states. Their alternation is regulated by an homeostatic control that works like a beam balance. Sleep-promoting molecules build up during wakefulness, shifting this balance toward the sleep state. At a certain point, these molecules are so many and the sleep pressure so high that the neurons that control sleep activates. When these neurons activate, they release neurotransmitters like GABA and neuropeptides that suppress wakefulness. However, an animal must be able to wake up even without an alarm clock, meaning that sleep must be also limited and controlled. Unfortunately, how this happens is not completely understood. Hi, I'm Lorenzo Rossi, a PhD student in Eric Bringmann's lab. Here at the SCNCB in Dresden, we study how and why animals sleep. To study the alternation between sleep and wakefulness and how this alternation is controlled, we turn to C. elegans. C. elegans is a small nematode that in nature feeds on bacteria on decaying organic matter, a source of food that by its nature it's unpredictable and can disappear in a matter of minutes. When a newly hatched larva of C. elegans faces a period of starvation, it can halt its development to foster all its energy and all its effort into survival. During this developmental arrest stage, sleep becomes more frequent the longer the starvation continues. For worms, sleep is crucial since it triggers the expression of protective genes that are needed for survival. For us, it creates a convenient model to study sleep biology. C. elegans has also a relatively small nervous system where sleep is controlled by a single neuron called RIS. When RIS activates, it releases the neuropeptide FLP-11 that induces sleep. However, how FLP-11 release leads to sleep is not completely understood. Neuropeptides like FLP-11 usually in C. elegans bind to a certain type of receptor called G-protein kappa receptor. So to study how FLP-11 induces sleep in C. elegans, we study worms that were lacking G-protein kappa receptor known to bind FLP-11. We discovered that one G-protein kappa receptor called DMSR1, when it's mutated, causes an almost complete loss of sleep. In general, when G-protein kappa receptor binds their ligand, they start in the cell a series of reactions that can have several effects on the cell. In the case of DMSR1, we discovered that it can reduce the activity of a neuron. However, DMSR1 is expressed in many neurons of C. elegans. So to pinpoint where DMSR1 acts to induce sleep, we decided to mutate the receptor in subpopulation of neurons, leaving it intact in the rest of the nervous system. Specifically, we discovered that when DMSR1 acts in cholinergic neurons, it induces sleep. When we mutated the receptor in this group of neurons, sleep was almost completely abolished. But interestingly, we saw that DMSR1 has also another important function that depends on its activation on RIS. RIS, as we said before, is the neuron that controls sleep. But when FLP-11 binds on DMSR1 present in RIS, it reduces its activity. This creates a feedback system that shortens the duration of sleep. To summarize, we discovered that the FLP-11 DMSR1 signaling pathway has a dual function on sleep. From one side, it induces sleep by acting on cholinergic neurons, and on the other side, can limit sleep by acting on RIS. This simple yet elegant system ensures that each sleep period is limited at its start. The importance of this discovery resides on the fact that over the evolution, sleep has been conserved. This means that we can draw parallels and similarities between simple type of sleep like that of C. elegans and more complex and evolved type of sleep like the ones of mammals. This means also that the FLP-11 DMSR1 signaling pathway can underlie similar dual mechanism of sleep regulation in other species, and that the self-inhibition of sleep-active neuron can be a conserved mechanism for sleep regulation that is worth to study further.